Hello, everybody. Welcome to Mind the Gap, Primary Immune Deficiencies, a continuously evolving story. Hosted by the Jeffrey Modell Foundation for this amazing week, World PI Week. World PI Week started in 1992 with a vision that Fred and Vicki Modell had to bring awareness, advocacy, education, and research to the world, because we're in it together, and together, primary immune deficiencies aren't so rare. They worked with Congress and the NIH to develop Primary Immune Deficiency Week, and as a result of it, much needed funding has evolved since the 90s for primary immune deficiencies. One of those wonderful investigators who is a great advocate and partner in primary immune deficiencies is Luigi Notarangelo. Luigi Notarangelo is the Chief of Immune Deficiency Genetics Section at the National Institute of Allergy and Infectious Disease at the NIH in Bethesda. He's authored more than 650 publications on molecular and cellular basis of inborn errors of immunity and the treatment of these disorders. He has contributed to the discovery of genetic defects underlying inborn errors of immunity and is responsible for many discoveries, many collaborations, and therapeutic interventions that are so important to treating our patients today. The wonderful part of this webinar today is it brings together colleagues, providers, and patients to learn about the ever-evolving story of primary immune deficiencies. Dr. Notarangelo is also a member of the National Academy of Medicine and the Association of American Physicians. It is my honor and pleasure to introduce Luigi Notarangelo to give this amazing talk and to celebrate World PI Week. I want to thank the Jeffrey Modell Foundation, Fred and Vicki, for their ongoing support. Dr. Notarangelo, it's my pleasure to send it over to you.
Thank you so much, Lisa, for the very kind introduction. It is actually my honor to be here today, and honor also Vicki and Fred for all what they've done from the very beginning to organize this PI week. So I'm going to share my screen at this point. I need actually someone to allow me sharing for the webinar. It says the host isn't allowing sharing. Still a problem.
Hi, Dr. Nutter. I'm turning the feature on right now.
Okay.
Right now. Think you should be okay now?
Yes, I think so. Yes, thank you very much. Okay, I suppose you can see my screen. Are you seeing in presentation mode now? Okay. So I slightly changed the title of my talk and added this Mind the Gap at the very beginning, because in fact, what I want to do today is not just mention about what has been happening in the PI field for now over 2 decades, but really highlight some of the gaps we still have to fill, which I think are very, very important worldwide. So let me just start by discussing introducing the education objectives. So I want to discuss the roots and consequences of diagnostic delay and how genomic testing and artificial intelligence may help address this gap. Review current practice parameters in the management of inborn errors of immunity, also after a very recent publication on that area. And also importantly, review development of novel therapeutic modalities and discuss how access to care remains an important barrier. So when we think about primary immunodeficiency and what happened in the last couple of decades, actually a lot of things have happened. Initially, primary immunodeficiencies were defined as conditions characterized by susceptibility to a broad range of pathogens. And now we know that actually the spectrum of primary immunodeficiency is much broader than that. You can have immunodeficiencies where only a few, or even in some cases, a single pathogen may be involved, but it's not just infection. You have autoimmunity, hyperinflammation, and even malignancies as presentation signs or key features of a variety of immune of immunity. Primary immunodeficiencies have been defined historically as defects that affect the blood cells. They are hematopoietic intrinsic, but now we know many immunodeficiencies that actually affect a variety of other cell types, including some that really don't affect blood cells, but actually affect other tissues. The diagnosis of primary immunodeficiencies for a number of years has been based just on clinical recognition and very few and simple laboratory abnormalities, with the idea that eventually when the genetics came into place, the idea was that a mutation in one gene would cause one specific disease. And now we know that mutations in the same gene may actually cause a variety of different conditions, but also mutations in a variety of genes may cause exactly the same phenotype. And finally, from a therapeutic standpoint, for many, many years, the only thing we could provide was supportive therapy, antimicrobial prophylaxis, intravenous immunoglobulins. And then cellular therapy came into place in 1968 with the first successful bone marrow transplant. And more recently, we have gene therapy, but also precision medicine, which targets specifically the biochemical defects that are characteristic of some forms of immunodeficiencies. But as I mentioned, in spite of all of this progress, a variety of problems remain. If we look at the natural course of the disease, we may distinguish basically three different periods. There is an asymptomatic period, which may be variably long depending on the nature of the immunodeficiency. And then eventually patients will develop symptoms of the disease. So that's a symptomatic period. And if this goes on and on and on, and no treatment is provided, the patient eventually will experience disability, organ damage, and unfortunately, in some cases, die because of the disease. So we have ways of intervening on this natural course of the disease. If we are thinking of the asymptomatic period, which basically goes anytime from birth until the very early signs of primary immunodeficiencies, we have ways to make a very early diagnosis. Of course, we can have prenatal diagnosis, but now, very importantly, we have newborn screening available for severe combined immunodeficiency. We'll discuss also other forms of newborn screening we can do. And in case there is a positive family history, we can immediately look for the disease in the newborn simply by screening for the same disease that was present in the other affected family members. When the symptoms already appear, we can still do a symptomatic diagnosis as soon as possible once a symptom happens. But if we actually protract this later in time, and so there have been many years of symptoms, and so you delay diagnostic and therapeutic interventions, this is where actually you experience clinical morbidity, late consequences of primary immunodeficiencies, and unfortunately death in many cases, or premature death. So diagnostic delay there are a number of papers that actually report on the diagnostic delay, and these are just some of the figures that I took from relatively recent publications. Estimates of diagnostic delay in common variable immunodeficiencies has been nearly 9 years. In immune dysregulation conditions, 5 years, and in primary antibody deficiency, 9.5 years. So that's a lot of years, and many things may happen in that period of time. What are the reasons for this diagnostic delay? Well, what the Jeffrey Modell Foundation has tried to do now for many years is actually to improve awareness. And actually, this is all about the primary immunodeficiency week is about. So improve awareness of these conditions. Undoubtedly, the rarity of the disease doesn't make things easy because physicians may not be experienced with the disease, may actually have problems recognizing the disease, making a correct diagnosis. In many cases, multiple organs are involved, so there are multiple symptoms which result in a diagnostic odyssey for the patients. They have to go through multiple specialties, and when they go through multiple specialists, it's difficult to bring things together and come to a final diagnosis. We still have poor access to care for many patients worldwide, actually. And lastly, I want to again emphasize the phenotypic heterogeneity of immunodeficiency so that even defects in the same gene may result in quite variable clinical immunological phenotypes. And along with the poor access to care, in many cases also we have limited access to advanced diagnostics, which now has become very important, in particular when it comes to genetic testing. So these are a couple of examples from this paper from Canada, actually, where they looked at the number of subspecialties that have been involved in patients with primary immunodeficiency belonging to any of the inborn errors of immunity classification. And you can see here that on average, we're talking about between 5 and 10 subspecialties that have been involved, whatever the type of immunodeficiency was actually in place. And this resulted actually in a significant diagnostic delay now measured here in months. So you can see that on average, you have anywhere between 2 up to 7 years of diagnostic delay. And obviously, as I mentioned before, this translates, unfortunately, in higher risk of organ damage, irreversible organ damage even. So this is another example where multiple collaborators from the US looked at medical records, and they basically, in a national claim database between 2007 and 2021, they looked at over 1,400 individuals who were initially given a diagnosis of unspecified immunodeficiency. So the physician was aware that something was wrong. They couldn't really figure out what type of immunodeficiency was in place. So they make this diagnosis of unspecified immunodeficiency. And then they followed basically over time until actually the first diagnosis of a clear inborn error of immunity was made. So the median diagnostic interval in this case was over a year, but you can see here that in some cases was way beyond 2 years. And there were already in this paper some interesting observations that some underserved populations are more at risk of this diagnostic delay. So this diagnostic interval was 14% longer among patients living in predominantly non-white neighborhoods, despite the fact that this individual had more severe symptoms at the diagnosis of unspecified immunodeficiency. And they also had to go through multiple health encounters for pneumonia and sepsis in a year after this diagnosis. Residents living in areas with greater deprivation also had more severe inborn errors immunity symptoms and required greater healthcare utilization in the year after the diagnosis of unspecified immunodeficiencies. Now, you may think, okay, 1 year of diagnostic delay may not be too long, but keep in mind that a longer interval also translated into delayed intervention, in this case specifically with use of immunoglobulin replacement therapy, that these patients had already received a diagnosis of unspecified immunodeficiency here, which means that they had a previous history of disease that lasted maybe in several cases for years. So the symptoms typically precede the diagnosis of unspecified immunodeficiency by many months and years. And finally, most of the patients in this study had private insurance. And when you have private insurance, you're better served. But if you have a public insurance, Medicare, Medicaid, then you are less likely to experience such a short diagnostic interval. So what are the consequences of this diagnostic delay? I already mentioned organ damage. And obviously, that also translates into decreased quality of life. But there are other consequences of this diagnostic delay. Patients often receive inappropriate treatment because, again, the physicians are not sure exactly what the problem is. They try multiple medications, and every medication comes with its own toxicity. So you may have actually toxicities as a result of the treatment rather than a disease. There is economical burden that is associated with diagnostic delay just because patients have to go through multiple visits, multiple interventions, multiple investigations. There is impediment to definitive cure because anytime you delay your diagnosis, you're also making less likely the patient may eventually receive definitive cure, because if the organ damage is too severe, then definitive cure may not even be possible, and that obviously may facilitate death. So the consequences of this diagnostic delay, once again, are even more important among patients from underserved populations. You have one gap immediately right there, that the underserved population need attention for this problem. Here's another example of how underserved populations are a detriment for a disadvantage for overall survival. And so this is looking now at the risk of mortality due to an inborn error of immunity in individuals, less than 65 years, but even less than 24 years. And you can see here that the blacks versus white the American Indian and Alaskans versus white, the Asians versus white, Hispanic versus non-Hispanic. They're all at increased risk in each of these two age categories. And if you look at what immunodeficiencies they are particularly at risk of dying, actually the vast majority of the groups of immunodeficiency are here in this in this triangle, where actually there is higher mortality and higher prevalence among the Black population compared to the white. Only a few others actually are less on the other side. So even when it comes to definitive cure, and this is a study that we did with the Primary Immunodeficiency Treatment Consortium, looking at the impact of stem cell transplantation over decades in terms of overall survival for severe combined immunodeficiency. And here we're looking basically at the risk of dying following a bone marrow transplant for SCID. You see that here, the Black African American, the Asian Pacific Islander, they do less well than the white. And so again, even when there is definitive cure available, some populations are at disadvantage over the more served, better served populations. So how can we close this gap? How can we facilitate, in particular, early diagnosis? Well, one way of doing this, again, this is something that the Jeffrey Modell Foundation has done for many, many years, is to improve awareness. Another one that also the Jeffrey Modell Foundation has pioneered is to develop artificial intelligence and machine learning algorithms mining electronic health records. And finally, develop more affordable and more powerful diagnostic tests, which includes a widespread and affordable genetic testing, developing a network of laboratory centers that are dedicated to identify inborn errors of immunity, but also creating a network of specialized referral centers. So let me go through this very briefly. You are all familiar with the 10 warning signs that the Jeffrey Modell Foundation established many years ago. And then they came up with an adjournment of these 10 warning signs, including warning signs that were specifically for adults. And there are some differences, of course, between the two of them. And then they developed this software for the recognition, intervention, and tracking of primary immunodeficiency. This software, the Spirit, which mines electronic records, matches 1,400 ICD-10 codes and over 17,000 pharmacy codes to the 10 warning signs. That actually help identify subjects with primary immunodeficiencies. And as a result of this, actually, this other study, which was published a couple of years ago, looked at electronic health records of 152 million United States patients, and that included a cohort of 41,000 inborn error of immunity patients with at least one verifiable diagnosis of inborn error of immunity that had been established at least 2 times compared to a matched set of controls. And the objective of the study was to quantify the value of having at least 2 warning signs as an indication of a potential diagnosis of a primary immunodeficiency. And what happened, in fact, was that proportion of inborn errors of immunity with over 2 or more warning signs was higher than in the controls. That's exactly what we wanted. But some warning signs, specifically those related to infections, were more heavily weighted among the inborn error of immunity cohort. That means that also we need to go beyond the 10 warning signs and include additional features that help us recognize primary immunodeficiency. What are these features? Well, I mentioned in one of my first slides that actually autoimmunity and immune dysregulation has now been recognized as a key feature of a variety of primary immunodeficiencies. So we need that incorporated into the warning signs. And so this is something that, for instance, Pérez Soler Palacín has tried to do in Spain. So he created this PIDCAP, which basically means to improve the inborn error of immunity recognition power of warning signs. It has been created in the Catalan health system, it is in Barcelona, and this is an expert-based algorithm weighing an extended version of the 10 Jeffrey Modell Foundation warning signs based on the ICD codes. And then this is integrated into an electronic health record software to alert primary care professional of the risk of developing a primary immunodeficiency for individual patients. And there are actually different signs that have been developed for the pediatric and the adult cohort, pretty much like the Jeffrey Modell Foundation did. But now you see here that autoimmune diseases, systemic endocrine autoimmunity are included, cytopenias are included in these warning signs. So that improves actually the capacity to recognize some forms of primary immunodeficiencies. I mentioned that artificial intelligence and also machine-based algorithms may help actually when analyzing structured electronic record, doing electronic health record, text mining, and claims data, help predict basically which patients may in fact have a primary immunodeficiency and improve the diagnosis, reduce the gap, reduce the delay in diagnosis that many of these individuals experience. Eventually, what you would like to do is once you identify potential patients with primary immunodeficiency, you want to confirm that. And that means actually you need to have laboratory tests that actually are final confirmation of the diagnosis of primary immunodeficiency. And what would these laboratory tests be? Well, one way of doing this, and has become now very popular, is of course genetic testing, so DNA sequencing, but you also have other advanced laboratory tests. And so let me go through these very briefly. First of all, the Jeffrey Modell Foundation, once again, years ago, understood the importance of developing proper testing for primary immunodeficiency, and they defined 4 different stages of testing for primary immunodeficiency, from the more simple to the more sophisticated. And then they also allowed basically genetic testing by making genetic testing available to a number of individuals across the world, actually, so that they could take advantage of advanced diagnostic testing, whole exome sequencing, and now even whole genome sequencing, and be therefore screened for a variety of primary immunodeficiencies in a more sophisticated way, a more powerful way. Very recently, this publication has been produced, and this is actually a publication that really tries to give practice parameters for diagnosis and management of primary immunodeficiency. And Dr. Forbes is actually one of the authors of this paper. And the idea is, in this case, actually is to look at diagnosis. And so how to make a diagnosis in front of a suspicious clinical history for inverness of immunity. And these are the tests actually, they're basically recommended with different levels of evidence. What they look at here is the certainty of evidence that this test, actually this working up, is efficacious, and also the strength of the recommendation, how strong or how conditional is this recommendation. And you see here initial workup and also targeted testing, pretty much like I showed in the previous slide, going from the more simple to the more sophisticated, the more focused on individual forms of primary immunodeficiencies that you can see here. So it is important to accelerate the diagnosis of inverness of immunity, and how to do this so that we can go from early identification symptoms and laboratory confirmation to even pre-symptomatic detection. And when it comes to pre-symptomatic detection, of course, our mind goes to newborn screening, which has become available for babies with SCID, universal newborn screening in the United States. As of December 2018, all 50 states and territories are screening for severe combined immunodeficiency in babies, simply analyzing blood that is collected on a Guthrie card at birth for a variety of disorders, including now SCID. So all 50 states are screening. And with the Primary Immunodeficiency Treatment Consortium, we looked at what is the impact of the newborn screening on survival after bone marrow transplantation for SCID. Because the background here is that in spite of all of the advances in advanced care, in improved technologies to do a bone marrow transplant, improve the treatment of graft-versus-host disease, as a matter of fact, the overall survival for babies with SCID that have been transplanted in the 80s until the 2009 had not changed. You see here, these 3 curves are absolutely overlapping with each other. And the only difference was observed after 2010. And this is exactly where newborn screening was being implemented in the United States. And in fact, if you look at what happened in this decade, you see here that the survival of babies that have been identified with SCID because of the newborn screening was actually higher than those that have been identified just because of clinical symptoms, mostly infections, as a result of their severe combined immunodeficiency. So there is no doubt that newborn screening has completely changed our perspective of cure in babies with SCID, perspective of cure and overall survival for life, actually giving these babies a chance of a normal life, both in terms of quality of life and in terms of duration of life. And so when it comes to, again, what to do in front of an abnormal newborn screening result, this publication identified what to do immediately. So how to confirm basically this could be a SCID, what tests need to be done, and what to do in cases where actually the screening for SCID comes back positive, but in fact we are not dealing with actually a severe combined immunodeficiency with other forms of lymphopenia, which is an important aspect not to be forgotten. So genetics obviously is now very, very important. And this slide shows the increasing number of genetic defects accounting for primary immunodeficiency that have been reported in the classification of the inborn errors of immunity between the 80s, from the 80s until the most recent classification was published last year in the Journal of Human Immunity. And now we see that basically we're dealing with 559 diseases resulting from variants in over 500 genes. But like I mentioned before, it's not just one gene, one disease. One gene may actually result in many different phenotypes. And there are many reasons for this. You can have loss of function or gain of function in the same gene, resulting in completely different phenotype. You have some gene defects that actually have a dominant negative effect, and so suppress even the activity of the wild type copy of the gene. In some cases, having one copy of the gene and not the other copy is enough to have a disease. And in some cases, the mutation actually introduces new dysfunction, new function, and new dysfunction to the gene product. You may have a different functional impact within the same disease mechanism. So different degrees of loss of function, for instance, may result in quite different phenotypes. You have some cases where we all have, two copies of a gene that is on a non-X chromosome or non-Y chromosome, but only one of them may be expressed. So this is called monoallelic expression. That may result in a different expressivity of the disease within various family members. And you may even have mutations that are not present in our germ cells. So they're not present in all of the cells of the body, but they only appear in some cell types, in particular in some blood cell types. And so then you have what is called a somatic mutation, a disease that happens at some point in the life of the individual. And on the other hand, you may be individuals who were born with a genetic disease, and then you have a reversion of the mutation in some cells. And so you may have some changes in the disease phenotype. So a variety of different confounding factors in interpreting the genetic results. So obviously, it would be nice to have a way to identify genetic defects in all newborns, not just in those with SCID, but in all forms of primary immunodeficiencies, whatever the disease is. But there are actually reasons to think of these. First of all, as I said before, there is disease heterogeneity. We are now dealing with nearly 600 forms of primary immunodeficiency. So it's not just SCID. SCID only is a dozen of different diseases. Now we're talking about 600 diseases. You can't really screen for all of these primary immunodeficiencies at the same time using simple biochemical markers. So there is some, this is beyond our capacity. The vast majority of the primary immunodeficiency do have a genetic basis. So if we had a way to look at the genes at birth, we might actually identify many, many forms of primary immunodeficiencies. And indeed, next-generation sequencing provides a single modality which enables parallel screening for hundreds of disorders, which differ in terms of clinical phenotype and disease-specific biomarkers. So in theory, there is a strong rationale for applying next-generation sequencing to identify all forms of primary immunodeficiency at birth. How practical is this? Well, there is already an experience, and this is actually happening in New York, where 6 hospitals are screening now, are offering all genome sequencing to all newborns. The first study was published last year on 4,000 newborns that have been enrolled in 6 New York hospitals. I can tell you now that the number has increased to 20,000. This was a diverse population, as you can see here, including various races and ethnic groups. The idea was to look for mutations in a number of conditions that actually have established intervention. So you want basically to make a diagnosis where you can offer something to these patients, and that includes 156 early onset conditions with well-established interventions, but also included, and this was optional for the families to accept or not to accept to be enrolled in this study, 99 neurodevelopmental disorders associated with seizures. Now, of these 4,000 individuals, 147, which is 3.7%, tested positive for 151 conditions, and 120 of them were true positive. Among them, there was one case of a male who actually had a gamma chain mutation. Gamma chain is the gene involved in X-linked severe combined immunodeficiency. Why is this important? Because this male had actually passed the newborn screening. He had a normal newborn screening, normal levels of T-cell receptor excision circles. He had actually a fairly normal, not quite normal, but fairly normal number of T cells, and these T cells were diverse, but actually the T cells were unresponsive to cytokines like IL-2 and IL-7, which is typical of mutations in a common gamma chain. And so that actually was predictive of a deleterious mutation. In fact, we do know that this is a deleterious variant. And so the baby was transplanted at age 4 months and has been cured. And hadn't we had this available, he would have gone and developed symptoms of the disease and maybe would have suffered from irreversible organ damage. So there is another study that is now being funded by the NIH. It's called Beacons, Building Evidence and Collaboration for Genomics in Nationwide Newborn Screening. Again, the idea is to apply universal newborn screening through whole genome sequencing for a variety of conditions. I'm not going to go through the methodology, but just to emphasize that, again, we want to focus on what we call actionable conditions. So these are situations where there is an intervention available or there is a very strong surveillance strategy that can be initiated in infancy and is expected to meaningfully alter the medical management and improve outcome. There are approved treatments or published surveillance guidelines to begin before 1 year of age. There must be a diagnosis supported by non-genetic confirmation test or characteristic findings of the disease. And finally, the variants that have been identified need to be identified as pathogenic or likely pathogenic. And so this study, which has been funded again by the NIH, will eventually screen for 777 conditions, which includes 743 genes, one condition which is associated with DiGeorge syndrome, the 22q11 deletion, and 2 aneuploidies, including trisomy 21, which is another cause of low T cells at birth quite often. And this is how Beacon's newborn screening compares to the Guardian study that I mentioned before, and even to another study also applying whole genome sequencing, which is called Early Check. And so again, what is important is that many of the conditions that will be targeted by the Beacon study are in fact related to primary immunodeficiency. So there is great hope that actually this novel diagnostic strategy, very powerful diagnostic strategies, will enable to identify many more patients with primary immunodeficiency very, very early in life, in fact, at newborn age. But there are still some challenges. First of all, as I mentioned before, the diseases that we want to target must be actionable. So these are diseases basically where we must have an intervention or a preventive strategy available. But we do know that in some cases, the disease may have actually a variable onset in terms of age. And so do we want to target also those that actually may appear later in life, or do we want to forget about those? Do we want to identify only those for which we have a cure, or also those in which we can provide meaningful supportive care? There must be standardized approaches for the management of infants with a positive newborn screening, and yet these standardized approaches have not been established yet. There is still a lot of variability in the access to diagnostic services and specialized treatments across countries worldwide, but even within the same country and even within the United States, among different states and territories. And finally, the evaluations of long-term clinical outcome and economic implications of this program remains to be assessed. one of the important aspects is that once we identify a variant in a given gene, how do we know that that variant is indeed disease causing? And, of course, the nature of the variant, is it a variant that actually prevents expression of a protein, or is it a variant that would allow expression of a protein which one single amino acid difference? I mean, that makes a difference. In some cases, we do know that that variant was seen before in other patients with the disease, so we're more likely to think that it is indeed disease causing. We may interrogate population-based public databases in the healthy, in the general population. We may use predictive tools. And finally, we have functional studies or animal or cellular models available in some cases to identify, to establish whether the variant is pathogenic or not. But in many cases, actually, that variant may be identified for the first time in a single case in a new gene. And so that makes things very, very difficult to assess. And that's an important aspect when the one has to consider in the development of these whole genome sequencing studies for the newborn population. So there are, in fact, tools that have been developed which have their own strong power. And one of them is called alpha-missense, which is now broadly used. And this actually incorporates many different things, structural aspects of the protein and how the protein structure varies as a result of individual variants, what is the frequency of that variant in the general population, AI models. But even that actually has its own disadvantage, because there are some situations where even this powerful tool, which is publicly available on the web, does not always predict properly whether a given variant is indeed disease causing or not. And there are many, many different reasons for these, which are intrinsic to how alpha-missense is constructed. So perhaps there is a need also to couple these predictive algorithms with functional assays. And the importance of doing these has been clearly shown by the group of Mike Hirschfield looking at adenosine deaminase deficiency, where in this study, a very recent study, we actually looked at what is the impact of alpha-missense, but also functional studies measuring exactly levels of metabolites and predicting actually whether the disease, the variant is pathogenic or not. And so you looked at a number of variants in the adenosine deaminase gene and classified them into 4 different groups. These ones in red are the ones that actually have the lowest activity. So these are the most deleterious. But these ones here, they retain robust activity. And so one may wonder, are these pathogenic or are they not pathogenic? And then he looked how they actually segregate into a disease that presents very early in life. These are babies, kid babies, that are already suffering from severe infections very early in life. And you can see here that this population here is enriched actually in variants that are belonging to group 1. So they are indeed the variants with the lowest activity. And by contrast, you see here that individuals who had this kind of variants here had higher level of adenosine deaminase activity, and the toxic metabolites in the red cells were actually rather low in terms of levels. And then he looked how alpha-missense, was alpha-missense able to predict actually this different behavior and distribution of these variants? Well, up to a point, because alpha-missense had been correctly identified variants that were likely benign, as you can see here, and then actually low alpha-missense score correlated with higher levels of activity. But you see here other variants that actually alpha-missense are predicted to be very deleterious, and actually they retain quite significant activity. So even these algorithms, they're not precise. You need a validation, you need a functional test to support the pathogenicity as predicted by the algorithm. And so what do I want to say? I want to say that we are in need of integrating a bunch of different information together and making that information available to clinicians worldwide. And this is the purpose of ClinGen, which is trying to offer a solution interpretation of genetic results. It's an NIH-funded centralized resource, which actually has primary immunodeficiency as one of the main areas of application. And these are the experts that actually have established ClinGen for primary immunodeficiencies. I'm one of the members of the SCID, a gene curation panel where we look at individual variants in SCID genes, and we help assess whether the variant is considered pathogenic or not. So I think it's important for the clinicians to have this instrument available. Again, this resource is available freely on the web. And these are actually some of the diseases that actually are targeted by ClinGen, but yet there are many other primary immunodeficiency genes which are still not curated. And this includes the chronic granulomatous genes, the TLR toll-like receptor genes, the STAP molecules, Mendelian susceptibility of mycobacterial disease, herpes simplex encephalitis genes. And these are only assessed in cases for dosage sensitivity, but not really curated in terms of the value of individual variants and whether they are pathogenic or not. So finally, what about management? And we have different ways of manage inborn errors of immunity. I talked about preventive measures, which include antibiotic prophylaxis, immunization, screening for complications, minimizing exposure to ionizing radiation for patients who have DNA repair defects. There are supportive therapies like immunoglobulin replacement therapy or use of immunosuppressive agents for patients with severe immune dysregulation. There are disease modifiers and targeted therapies, including small molecules, biologics, or adenosine deaminase replacement therapy. And finally, there are curative therapies, which really offer the perspective of fully curing the disease, including stem cell transplantation, gene therapy, or thymus tissue implantation. And again, that practice parameter publication that I mentioned provides some guidance here. How strong is the evidence for general monitoring, and which measures in particular of general monitoring, how to monitor for common morbidities, and also, what is the role of antibiotic, antimicrobial prophylaxis in general in a variety of primary immune deficiencies, how to prevent infections, looking at a variety of strategies, looking at the environment, use of blood products or vaccination in patients with primary immunodeficiencies. But another important aspect is, where do we want to do all of this? Because patients have a right to have their treating physician being able of taking good care of them. But at the same time, in many cases, there is a need of a referral center for specialized intervention. So this is actually, again, the same study that I mentioned before by Soler Palacine, who looked at what is the advantage of being treated at a local hospital versus going to a referral center. And there are plus and minus, as you can see here, for each of these different situations. And so it's important, basically, if possible, to integrate, bring together a care nearby, which is actually what the patients would like the most, of course, with proper referral and proper input from experienced treating physicians. And this is exactly what the Jeffrey Modell Foundation, again, tried to do by developing a consulting immunology, by finding this program here, find an expert immunologist. And similarly, the Immunodeficiency Foundation has created this consulting immunology program. European Commission has something similar, not quite exactly the same, but it's, again, another important resource. And finally, the Clinical Immunology Society has this primary immunodeficiency listserv, where actually the physicians are able to post individual cases and ask questions to their colleagues to get expert opinion on how to manage, how to address a specific clinical problem. Finally, in terms of management, again, what has been changing is the advances in definitive therapy, again, hematopoietic stem cell transplant, gene therapy, or thymus tissue implantation, but also precision medicine. And with regard to that, actually, we know that there are a number of immune dysregulation symptoms that may happen in patients with primary immunodeficiency. And we know now of many different targeted interventions. These are all monoclonal antibodies or small molecules that actually target all of these molecules here. But on top of that, there are also small molecules like, here for JAK inhibitors, for instance. And so the problem here is that on the one hand, we have actually very powerful agents to interfere with individual pathways that are compromised, dysfunctional in specific forms of primary immunodeficiencies. At the same time, these are rare conditions, and so companies are not willing to invest money to look at the efficacy of these interventions specifically for primary immunodeficiencies, because there would be no capacity to run a sufficient number to treat a sufficient number of patients and be that also economically advantageous to the company that is investing money. So here's what Dr. Forbes Sutter is doing with Troy Torgerson using what is called a basket trial, where individuals with different forms of primary immunodeficiency may benefit from the same molecule, may actually receive the same treatment. So the idea is actually to increase the patient population, pretty much like has been done in cancer, where the same drug may be used for different type of malignancy. So that's one way forward to make these advanced treatments available also for patients with primary immunodeficiencies. And obviously, the other major advance has been in gene therapy, which initially started with lentivirus, actually even before that, retrovirus mediated gene transfer, and now it's done mostly with lentiviruses. And here are actually the diseases for which there are clinical trials available using lentivirus mediated gene transfer for different forms of severe combined immunodeficiency, Wiskott-Aldrich syndrome, 2 forms of granulomatous disease, leukocyte adhesion deficiency type 1, and IPEX. But beyond this, there is now even a novel way of doing gene therapy, which is actually doing a real correction of the gene defect by doing either base editing or prime editing using the CRISPR technology. And these are actually diseases that have been already treated in individual cases by either base editing or prime editing. Yet, there is a major problem, that these are costly, that the companies, again, initially invest money and then they withdraw their funding just because it's not economically advantageous to them. And so there is a need actually to make gene therapy and access to care available to all. And these are 2 editorials by Alan Fisher, actually, in both cases, where he really calls for gene therapy being something that should really be available for all. And he actually is insisting that we need to think in a different way of making gene therapy possible, probably not just relying upon the companies, because the companies have to do their job. And of course, they have to look also into their own budget, but bringing together, the legislator, the taxpayers, and altogether policymakers and patient advocacy groups, and all of these working together to develop innovative solutions to ensure that access to gene therapies is truly equitable. It is another study by the group in Milan, in particular Alessandro Aiuti and Luigi Naldini, which identify the problems, I'm sorry for this Italian here, but the problems associated with gene therapy, high production cost, obstacles, and inconsistencies in the regulatory approval processes, inadequacy of cost-benefit analysis, and then feasibility and sustainability, and possible solutions to each of these problems. Well, let me finish by saying that significant progress has clearly been made in the characterization of primary immunodeficiencies and also in the development of more effective diagnostic and therapeutic modalities, but yet important gaps remain to be addressed to reduce the diagnostic delay, facilitate access to care. These gaps are even more significant in patients belonging to underserved population. We need to bring together the mining of electronic records, the proper laboratory tests, and genomic studies, because that may really change our way of facilitating an early and, in fact, definitive diagnosis. In particular, whole genome sequencing in newborns focused on actionable conditions may represent a transformative approach to early diagnosis and intervention. But again, we need to look carefully at its long-term clinical and economic implications. Patient advocacy groups like the Jeffrey Modell Foundation remain fundamental partners with physicians in promoting awareness and education and in advocating for improved access to care with health authorities. So a change may just be around the corner. And I'd be happy at this point to take any questions. Thank you for your attention.
Gigi, that was an amazing tour de force. Thank you so much for highlighting so many aspects of what we face and what we need to do to mind the gap, but also what we've done to get ourselves here. So
Thank you.
amazing. And, it's really about research and clinical and kind of marrying that pipeline together where, everybody from the patient advocacy groups to the clinicians to the physician scientists and for the PhD researchers really all work together on the same page. The Jeffrey Modell Foundation supports that through the cycle grants that they do, through the Genie grants, the single gene defect, supporting the Vicky Modell Innovation Award at the CIS. They've really given us an opportunity in our field from an education and advocacy standpoint, as well as a basic science research.
Kudos to the Jeffrey Modell Foundation.
Yes, it's wonderful. We've both been benefits of that for sure.
Yes.
We have had over 400 registrants from 63 countries. Almost 200 people were able to join, an amazing feat. So I want to thank you. So we should get to some of our questions. One of the questions in the chat asked about it seems to have come from a patient who asked, as an adult with CVID, should they have genetic testing? And if they don't have, I'm going to paraphrase, the resources to get the genetic testing, what kind of things are available to them to make sure that they're adequately advocating for themselves and supporting that need for genetic testing?
Well, it's an important question. It remains true that the majority of cases of CVID remain genetically undiagnosed at this point, but things are evolving. And so we're also interrogating now different areas of the genome, not just the coding sequence of the genes. And we don't know whether there are variants in regulatory regions that may be involved. And there are cases actually of CVID or what was thought to be CVID in adults that actually turned out to be different conditions. We've had many cases like this. I had several patients with drug deficiency were initially diagnosed with CVID. And when we did genetic testing, we found that they actually had a combined immunodeficiency, as a matter of fact. So it is worth looking for a genetic defect also in adults with CVID. The yield is still relatively low in terms of capacity to identify an individual gene defect, but it's something that should be tried in any case. And I would say, again, the Jeffrey Modell has been really helpful in making genetic tests available for individuals who come from countries where genetic tests are not, widely available, or for families that cannot afford sustain a cost of genetic testing. The costs are going down. That's a good thing. And so I expect this to be more and more easier and easier, with time. But yes, I mean, there are ways to do this, and I would encourage actually doing it.
I think that's a great, that encompasses a lot of the questions in our chat. There's a few what seems to be patients that are joining that says that their specialist didn't recommend genetic testing. So it's important to know that there may not be a genetic test available or discovered, but it's important to check. And an answer that doesn't reveal itself now doesn't mean it might not reveal itself later. So, that's important to note. Along those lines, the non-infectious complications of immune deficiency has really kind of made the immunologist and the subspecialists really have to think broadly outside of their scope of practice. So do you want to talk about how we can, make that lens a little bit smaller in our ability to really identify and what tools are out there for patients and providers to grasp the really large context of these non-infectious complications?
Well, I would say yes. Actually, you are a master here because you have you have pioneered actually diagnostic approach to patients who may have immune dysregulation as a key feature. And immune dysregulation may come in different flavors. You may have true autoimmunity, which is not so rare in immunodeficiency. In fact, actually, even talking about CVID, ITP is immune thrombocytopenia is very frequently the presenting sign of common variable immunodeficiency. But it's all kinds of autoimmunity, cytopenias, organ-specific autoimmunity. But then you have also inflammation, and you have hyperinflammatory manifestations, granulomas, for instance, that are often seen in patients with primary immunodeficiency. I think immunologists are now better trained in identifying and recognizing these conditions. It's not just infections. So again, I would say if patients experience this kind of problem, I think their immunologist should be the primary point of contact. They are the ones actually who have a better understanding of these problems and may be the ones who actually can identify which are the best ways of investigating such defects. Again, some of these tests are not broadly available. They require specialized centers. But again, immunologists will know who to go to. And I'm sure, even for patients living in countries where these tests are not available, we as a community are trying to help, patients wherever they are. So, just reach out to your physician, your immunologist will reach out to us. So we work together.
Yeah, I think that's great. There was a really good question in the chat, and I think you kind of touched on this in your talk. Is there a way to fast track the diagnosis using a method other than genetic testing by a biomarker? And so, when is that right time to get genetic testing versus a biomarker versus both? It's almost like a chicken egg thing, right? So,
Yeah. So, well, unfortunately, we don't have simple biomarkers that cover all forms of immunodeficiency. If you do genetic testing, if the genetic test was able to identify all forms of immunodeficiency, it really would. I mean, because then, we are our story is written in our genes to some extent. So by finding a genetic abnormality, you know exactly what you're dealing with. With biomarkers, you have individual biomarkers for different groups of diseases. Again, these biomarkers may or may not be easily available, but they wouldn't cover the whole spectrum of primary immunodeficiency. But that said, when you have a clinical suspicion of a group of disorders, then you may use biomarkers, and biomarkers become even more important or equally important as genetic tests, actually, because they really validate the result of the genetic test. They really substantiate what the gene defect has shown. So I would say, again, your immunologist would know which biomarkers to use and who to go to.
So I'm going to paraphrase someone from the Shwachman-Diamond syndrome who is asking about genetic testing when there's a nearby pseudogene. So when there's a disorder where the genetic testing isn't really straightforward and maybe whole exome and whole genome sequencing might not pick it up, how do you advise providers or patients to advocate for the alternative ways to get genetic testing?
Well, actually, even in that area, there is an evolving field which is represented by long read of DNA. And the long read of DNA are able to address the issue of the pseudogenes. So they can really distinguish between variants in the gene versus the pseudogene. They can also identify structural variants. Are these broadly available? No, they're not. They're still limited to a few centers. So again, this requires interaction between immunologists and the geneticists and between geneticists among themselves, because again, these assays are not broadly available to all genetic groups. So, but these things are evolving. So again, I would expect major changes in terms of affordability and availability of these tests in the next few years, really.
Yeah. So Rosa Bacetta has a really nice question. There are many advanced lab tests that really help to understand the disease and the pathogenicity of the mutation. It's difficult to integrate these tests in medical records. How do you facilitate this, especially for those that are not available in a clinical certified lab?
Well, that's an important question indeed. And it's an important question, especially for countries like the US, right, where actually you have CLIA certified tests and then you have research tests. You can't incorporate research findings into the medical records. But as a scientist doing some of these tests, we maintain a channel open with the referring physician. And so you, even if you can't really make action based on the research test may help support hypotheses that have been raised by the treating physician. And if you put them together with other features of the disease, with genetic results, then it's still okay. But it's an important question, and I think we need to do better there to make some of the research tests available also in clinical as clinical tests in clinical labs. Again, there are too many of them, and some of them are not easily, amenable to becoming CLIA certified. But there is progress even there. So again, it's another area where I think we can expect some progress.
Thank you so much, Luigi. You have many accolades in the chat telling you what a wonderful talk. It's always a pleasure to introduce you and to interact with you. And I wanted to thank Fred for joining in the background. I want to thank the Jeffrey Modell Foundation and their wonderful leadership for thinking of this really important series. I want to thank all of you for joining, because it wouldn't be worth it if we didn't have you to talk to. And it's such a pleasure to be a part of this series and the programs that are happening in World PI Week. We hope that all of you can join us for future webinars. And I want to wish everybody a wonderful day and a wonderful World PI Week. Thank you, everybody.